All right, welcome to another installment of the Molecular ML Reading Group. Today, we are absolutely thrilled to have Sarah Alamdari present. This is the EvoDiff model, and she's coming from Microsoft Research. So thank you, Sarah, for coming, and take it away.
Yeah, thank you for the kind introduction. And I just want to add, feel free to interrupt at any time, or if you'd like to deep dive into something, feel free to stop me while I'm going. So yeah, today I wanted to talk about EvoDiff. It's this model that we recently released. It's a discrete diffusion model for controllable protein sequence generation. The motivation behind this, of course, being that proteins are these powerful actuators of biology. I think this audience is fairly familiar, but proteins are biomolecules that are on this length scale of 3 to 10 nanometers, so quite small. And they're responsible for precisely modulating nearly every cellular process. So in our body, we can think of on the scale of 100,000 proteins, which regulate the different processes in our body. And UniProt, this large database, has recorded on the order of 85 million unique protein sequences that exist. Now, what are proteins? Well, proteins are composed of these chains, these linear chains of amino acids, and they're comprised of really simple chemistry. So all amino acids contain this carboxylic acid group here, an amine group, and then what I've highlighted in red here is this side chain that composes the kind of chemically active part of an amino acid. And when we combine these in a chain, that's then what creates a protein. Now, this side chain R is quite interesting because all of this functional diversity that we can observe in our bodies or in this world due to proteins arises from just a few building blocks. So R in a natural protein can arise from the combination of these 20 different amino acid groups over some variable sequence length. So this can be from tens of amino acids to thousands of them. Inspired by this, the field of protein engineering is really motivated to develop protein solutions to these modern challenges. So how can we take inspiration from the way that these proteins actuate processes naturally and use them to solve new modern challenges? So for example, in disease, can we develop these drug delivery agents or antibodies? Can we use them as molecular tools? A famous example of this is the green fluorescent protein, which is used in a lot of imaging applications, or similarly, can we use them in industrial applications? So for example, in the field of environmental sustainability, people are using enzymes to degrade new biodegradable materials or sequester carbon through the use of proteins that can catalyze new reactions, so an enzyme. And what I'm really introducing to you is this idea of this sequence structure function paradigm, in that this arrangement of amino acids, which are given by these single letter codes, is what enables a protein's 3D structure and ultimately enables its function. Now, conventionally, we've seen a lot of these structure-based approaches to protein design in that you identify some function of interest. We use a generative AI technique to then identify and redesign the relevant structural components of this process. And then again, we can use some AI tool to then predict its corresponding sequence. And there's a few really exciting examples of this. So RF diffusion, chroma, and folding diff are all three really nice generative structure-based examples that have been used to even experimentally, in an experimentally validated sense, validate some of these generated protein structures, showing that this is an extremely powerful way to design new proteins. However, when we think about this process, what our group wanted to do was reassess this from a sequence-first based approach to protein design. In that, because we know that a protein sequence is what enables its structure and ultimately its function, can we do this by bypassing maybe the structure prediction step? So can we identify simply a function of interest and predict the corresponding sequence that can carry out that task? Now, why would we want to do this? Well, there's a few bespoke reasons. The first being that there are significantly more data available for sequences compared to structures. So here's a nice visual representation of the fact that many of these structure-based models are trained on on the order of 200,000 available PDB crystal structures available for proteins, whereas sequence-based models can exploit much larger data sets. So for example, we train on 50 million protein sequences available in UniRef 50. Similarly, many of these proteins don't fall under the classic structure function paradigm. So there's an entire class of proteins known as disordered or intrinsically disordered proteins that aren't captured in this structure-based scheme, but we know that are critical to both functions in the body and in many diseases. And lastly, this eliminates that step needed for indirect sequence design. So going from a designed structure to then some corresponding sequence. So this is exactly the question we asked in order to develop EvoDiff. And it's that, can we operate directly in this universal design space to build new generative models of proteins that are both performant and controllable? Here we develop EvoDiff. Evo here stands for the fact that we train over an evolutionary scale of data, and diff in that we're using a discrete diffusion model as the backbone here. And I'd just like to shout out all the team members really quickly who were involved in this project. It was a huge collaborative effort. Alex, Nicolo, Rianne, Ava, and Kevin are all at MSR as well. And Nitya really led a lot of the beginning phases of this project during an internship she had while an undergrad at Brown. And she is currently a grad student at Stanford doing very cool things. Okay, so just to dive in a little bit deeper into these diffusion models, if you're unfamiliar with this, the idea is that you take an input data and slowly corrupt it. I don't know if you can see my mouse. No, you can't. Going from right to left over a series of time steps. And this is a controllable corruption process until you end up with something on the left, here denoted by x of sub t, that can easily be drawn from a distribution. So typically we're corrupting to random noise. Now, to place this in the discrete context and start with a really simple example, we take a protein sequence here, and then in this OADM model, which the citation is missing. Oops. Okay, so we assess a few different models for this diffusion task. The first is known as order agnostic diffusion. This is a case where you take an input sequence, you slowly corrupt over all the positions in the amino acid sequence. In this case, we do so with a mass token until the entire protein sequence is corrupted. Your model is then tasked with predicting the mass tokens over the course of training and deciding what those mass tokens are. At inference time, you can then start with a fully masked sequence and then residue by residue uncorrupt in an order agnostic fashion the corresponding amino acids. Oh, there's the citation. This was originally developed by Rianne's student and presented at ICLR in 2022. The second approach that we looked at is called D3PM for short. I believe it's discrete denoising diffusion. I messed that up. Discrete diffusion and nope, denoising diffusion in discrete state spaces. There we go. And in this case, instead of corrupting using a mass token, we're corrupting according to some probability of a transition matrix. So now we're corrupting by mutation. So at each step, our transition matrix determines which amino acids are mutated to other residues with some probability that we can control. And then our task of our model at each step is then to uncorrupt or figure out what the true token should be at that corrupted location. This is a much more difficult task for the model as it doesn't have context in this case as to what is being corrupted. Whereas in the mass case, that's denoted by a symbol. We find that this OADM scheme performs quite well in this case. We did a bunch of benchmarks here, and so we'll denote this model as our EvoDiff-seq model. And just to reiterate, we train this over an evolutionary sequence space over many organisms and many functions. And the base of this model is a 640 million parameter convolutional neural network. And this is just a nice GIF of showing what happens at inference time. So again, this is just showing in that order agnostic fashion that we can predict each identity of each amino acid residue at each position. And it shows the corresponding prediction of that structure over here on the right. Now, if you're familiar with the field of machine learning and particularly these approaches to modeling protein language, this is quite similar to what we call a left to right autoregressive model, in that in an autoregressive sense, you're predicting the next token at each residue in the sequence. And so the OADM model is essentially an extension of this, where you're predicting every order at every step. The reason that we prefer this method is that what we find is that this autoregressive scheme is not amenable to a conditional generation, where you can provide it with some context at inference time and then generate around it. You are stuck in this left to right decoding order. This is also akin to a typical mass language model, like has been used in CARP or ESM, where you're just learning a fraction of the tokens and asking it to predict those. So you can think of our OADM model as being every order and every step, whereas something like ESM is only training one step at a time. And what we find is that exactly as we expect, the EvoDiff seq model can generate better from scratch, whereas in a mass language model, like in this case, you're not able to start from a fully masked sequence to then generate new protein structures. It's better at kind of filling in the gaps in this percentage of mass that it's trained on. Now, moving on, we wanted to evaluate the performance of EvoDiff in a few different ways. So here we look at the plausibility of our generated sequences, we look at their diversity, and then we ran some experiments on the conditional design as well. And first, I'll dive into the plausibility. Okay, so we adapt this scheme that's being used in the field of protein validation, and we take a generated sequence from our model, run it through some structure prediction. In this case, we're using OmegaFold. That gives us a PLDDT value, which is a confidence in how true this predicted structure is. We then pass this structure through an inverse folding software. In this case, I think we ended up using ESMIF. That gives us a predicted sequence, and we evaluate the self-consistency between the predicted sequence and the original sequence. Now, we can take our test set and pass it through this workflow. And here I'm plotting the perplexity, so the confidence in that prediction versus the sequence PLDDT. And as you can see, even on real sequences, this workflow isn't perfect, in that we get a good amount of protein sequences that fall in this high confidence region in the lower right, but there's also, this whole region of protein sequences that are not as confident. But we use this as a general metric to understand how our model's doing. Now, if we look at some of the generations from EvoDiff, we see that we follow this general trend in that we end up with some of these high confidence candidates and generally get scores around what we're expecting. Now, if we pull out some of these proteins from this high confidence range and visualize them, we get a really nice set of examples of proteins that are predicted to be realistic by this model at a variance of length scales.
Hi, Sarah. Can I ask a quick question?
Of course.
What was the rationale for using OmegaFold for checking these sequences as opposed to other structure prediction networks?
Yeah, it's honestly, it's just quite a bit faster. We have now, since this preprint coming out, validated a few of these with AlphaFold now and find that they work pretty similarly, but it was just more efficient for running like thousands of sequences at a time. We did a bunch of benchmarks that are in the SI, and it just, I think, was infeasible to try and run AlphaFold on everything.
Oh, I see. Thanks. And when you were checking AlphaFold, were you using AlphaFold with MSAs or in single sequence mode?
Yeah, so we would, we would use AlphaFold out of the box, in that we would give it a sequence, it would run a homology search, and then run through it with that generated MSA.
Okay, thank you very much.
Yeah. Okay, And so we can compare this to a few different benchmarks. I'll showcase 3 right here. The first is we compared it to the results of our left to right language model. This is the state of the art in protein generation. And we see that this does slightly better than our EvoDiff-seq model. The training for this is quite a bit more efficient than the one that we're doing here. So we expect that, given that we train these on the same data set for a similar amount of time. However, it's important to remember here, I don't think my graphic will pop up, the rationale behind using this EvoDiff-seq is in its conditional generation abilities. And second, we can compare this to the results from using something like ESM, and we see that it does significantly better at generating realistic proteins. Okay, and so now the next thing we wanted to look at was the diversity of our generated sequences. To do this, we take a set of our test sequences, run them through ProT5, which is a different sequence prediction model, get those embeddings, and then plot them in a UMAP space for visualization. We then generate a similar set of sequences, get those embeddings, and then map those on top of our predicted UMAP space. What this is doing is capturing generally just important biophysical properties related to shape, function, and otherwise. As we can see here visually, we have a good extrapolation of this space. I'm currently looking into kind of what these regions are that are not covered well by our model. But also we can numerically represent this using what we call this Frechette protein distance. This is just distance. It's just a Frechette interception distance between the 2 embedding spaces, where a lower number here is more desirable. Now, again, we can compare this to some of our key baselines. The first being that left to right autoregressive model, where we see both in terms of visual coverage, it's better, and the FPD, it is better. But again, we want that benefit here of the conditional generation scheme that's only capable in EvoDiff-seq. And second, we can compare this to proteins generated from a mass language model, in this case ESM2, and we see that we do significantly better than that model, which again we expect because this is quite outside the possibility of, what this model is capable of. And lastly, we also compare this to a structure-based diffusion approach. RF diffusion at the time of making this was kind of our state-of-the-art model. And we see that we do much better in covering the protein functional space than compared to RF diffusion. And again, this makes sense, and we can attribute this to the scale of evolutionary training. We wanted to do this again in terms of the natural secondary structure distribution. So can we interpret some embedding that tells us something about what the predicted secondary structure is of these generated sequences compared to our test sequences? So again, we walk through this workflow. We get a expected secondary structure distribution according to those test sequences, and we can map it here, where this is the helix dimension, this is our % of strand per sequence, and then you can imagine a third dimension here, which is on the disordered region. No, sorry, the beta region. Okay, and we can conduct this again for our EvoDiff-seq model and characterize this using a KL divergence between the distribution of our test and distribution for our generated sequences. And we get a distribution that looks quite similar. Again, when we compare to our left to right autoregressive and ESM2 models, we see that we're on par with the state-of-the-art method. We do better than ESM, and this will come up in a bit, but interestingly for RF diffusion, we find that we're able to generate more variety of secondary structure compared to these structure-based methods. So for example, this region here of low helix and low strand would indicate more disordered proteins, which is just simply something we're not able to observe with a model like RF diffusion. And lastly, this is kind of the exciting part of our model, but we really wanted to use EvoDiff to see if we could controllably generate diverse and plausible conditional designs, meaning that we fix some part of this subsequence and inpaint the remainder, and mask and decode the desired portion in some arbitrary order. So for this task, we look at 3 different tasks. The first is this idea of evolution guided generation, which I will introduce here quickly in that it involves MSAs. So for EvoDiff, we ended up training a secondary model that leverages evolutionary information. So we take a query sequence, we conduct a homology search, and construct an MSA that we subsample to contain 64 related sequences that are a max of 512 residues in length. Oh, sorry, I'm off with my animations. And then we pass this through the same corruption scheme that we described earlier for the sequence only. So we take an input MSA that might contain insertions or deletions. We mask out every residue within the MSA until we end up with something that is fully masked. And for this, we train a new model that's trained on 100,000 open-fold MSAs, again, covering this wide space of organisms and functions, but in this case, the backbone is a 100 million parameter MSA transformer. Now, what's cool about this is that we have then accidentally almost created this amortized local generative model, where our inputs describe natural classes of protein families and then are able to generate new functional proteins within that class. So we come up with this idea of evolution-guided generation, where we can exploit the fact that the inputs are of related sequences and use that to generate new queries without having to do any additional conditioning on the model. So I'll walk you through this procedure. We take an MSA of natural aligned sequences. We mask out that query sequence only, leaving the context that's in the aligned sequences, pass this through our new EvoDiff MSA model, and then generate a new query sequence here. The rest of this workflow is then quite similar to assess plausibility. We pass this again through our structure prediction, get some related structure, again, inverse fold it, get the predicted sequence, and map this to the perplexity versus PLDDT plots, which are giving us some sense of plausibility of these generated sequences. As we can see here, I've plotted the validation set for this model. It does quite a bit, the MSA data does quite a bit better on this model, and we attribute that to this data set being a little bit more structured than the UniRef 50 data set. And when we look at our generated sequences on top of this, we see that we do quite well in terms of performance. Now I'll quickly pass through some benchmarks here, but when we compare the average PLDDT distribution and compare that to we're comparing the distribution of PLDDT and perplexity to other models here. We see that we do better than our ESM MSA equivalent, and we do slightly better than a POTS model, which is just sampling according to the probabilities in that aligned MSA. And what we also want to do here is then take that original MSA, the original query sequence, pass it through some structure prediction to get some corresponding structure, and then evaluate the TM score. So really, we want to see how new is this generated, or how similar is this generated sequence in structure to its original query prediction. And what we see is that overall, our generations are much more similar than using something like ESM MSA, and significantly better than using something like a POTS model. And lastly, we just wanted to compare the sequence similarity in this original query compared to our generative query, and we see that on average, we're able to generate new sequences with similar structure. Now, to just give you some visual of what that looks like, at a various sequence lengths, we're able to generate new sequences, so meaning low similarity, with shared structure, implying that they may have shared function. And this is critical to kind of the next two parts that I'll get into. Now, one of the tasks that we wanted to really explore was this idea of sequence inpainting of disordered regions. So given a protein sequence with a disordered region, can we give it a new plausible intrinsically disordered sequence? Now, I want to quickly turn back to this example that we had given earlier, and it's that these structure-based methods really lack the ability to generate disordered structures, mostly because it's just not included in our data set. Whereas we know that EvoDiff-seq is capable of doing so, as well as EvoDiff MSA. So to test this, this is a similar workflow that we've been using, but we take an MSA, we take a sequence where we know the region of a disordered portion of that sequence, we mask out only that part, and then generate a new disordered sequence. We can do this again with our EvoDiff MSA model, where we can use a database of known aligned disordered proteins. They have some shared structural regions and shared disorder regions across many organisms, and conduct that same experiment where we mask out the query IDR, again, only working with the query here, leaving that context in there, and then generating a new intrinsically disordered region within a protein sequence. We can take that and pass it through some disorder predictor. Here we're using a model called Dr. Berg, not as well characterized as something like AlphaFolds or OmegaFolds, but works pretty well for these purposes. And the output of this is a disordered score for every residue in our protein sequence. And again, we can compare this to our original query and what that might look like, and compare what the disorder prediction score is for both of these sequences. And ideally, it's the case that our generated sequence will have a high disorder score in the same region of something that we masked out. And here are just two very hand-pulled examples of this. First, with EvoDiff-seq, our first model that's able to generate a new disordered sequence within the region of this protein that's disordered. And similarly with EvoDiff MSA, our MSA-based model, we're able to do something similar. And what's interesting here is we're even able to inpaint some of these insertions. So we actually end up increasing the length of this protein sequence here. And lastly, we can just do some analysis on this as Dr. Burt is not a robust analysis for predicting disorder. And we see that, compared to random, we do a pretty good job of this. We additionally include some examples in the SI. However, what we find is that while EvoDiff-seq is able to generate new IDR regions that are quite dissimilar from the original protein sequence, EvoDiff MSA tends to generate things that are quite a bit more similar. So in general, they're kind of something more akin to scrambling the sequence. Okay, and lastly, I wanted to get into this idea of functional scaffolding. So in this case, we're really wanted to assess the limits of how well we can generate new protein sequences that exemplify a given structure. And the motivation for this is that this goes back to kind of this introductory question where many functions we know are identified by a particular motif that's stabilized by a scaffold. I stole this from a paper of the Baker's lab, but we know that these functions like epitope presentation, receptor traps, active sites, or protein-protein interactions can be observed through these specific structural motifs that can be identified. And we wanted to ask, can we scaffold these motifs using just sequence by itself? So can we maybe visually interpret where these might bind and just use that information to generate new scaffolds? So for example, we can look at this protein. It's a binding site of compact calmodulin protein that looks like this. In green, I've highlighted the functional motif that we know carries some functional activity, and in gray is the part of the protein that we'd like to scaffold. So what we do is we keep this green region in sequence space, we place it randomly within a scaffold of some set size, and then we begin to pass it through the model to then inpaint each residue. as you can see here. Oops. And then when we look at what the resulting structure prediction is here, we see visually we end up with, quite this striking example of scaffolding, this structural motif, but in sequence space alone. So we're able to do this without, any context about structure needed. Now here I'm just going to roll through a few different examples, but obviously we've included many more in the paper. Here, EvoDiff seek generates on one PRW a few times. This is the example I just showed. EvoDiff MSA, we have this really nice example of this enzyme, which we've been able to scaffold some of these beta strands, and we've actually made a smaller enzyme, which is quite exciting here in this case. And we do this over 17 unique problems that were originally published in RF diffusion. The criteria for a successful scaffold, we try and mimic off the criteria of RF diffusion in that the generated structure must have an RMSD of less than 1 angstrom of that functional binding site, and it must have a score greater than, I think we ended up going with 70, but it could be 80. I have to go double check. And we count those as successful scaffolds over this number of 17 problems. RF shows that they successfully scaffold 13 of the 17. EvoDiff MSA on par also successfully solves 13 of these 17. And EvoDiff seek does a little bit worse in scaffolding only successfully 8 of the 17. However, when we look into what exactly these problems are in terms of the number of successes, we see some interesting things arise. So here I've denoted on the left the number of successes from our MSA-based model, and on the x-axis our EvoDiff seek model, and each of these denoted are unique, one of those 17 problems where at least we get one successful scaffold for each. What we find is that in general, our MSA model is quite a bit more effective than EvoDiff seek, as seen here. We can also plot our best EvoDiff model, so EvoDiff MSA versus RF diffusion, and we find that they surprisingly have quite orthogonal strengths. So it seems that problems that EvoDiff MSA does well on, RF diffusion does not do as well on, and vice versa. And interestingly, we have a few examples of EvoDiff seek that does better than EvoDiff MSA. And when we investigate why this is, we find that these are examples where we don't have a lot of homologs. So for example, the MSA constructed here might not contain the full 64 by 512 sequence that has been seen by our EvoDiff MSA model. So it's like quite outside of our training task. And then in general, we can look at the structural similarity of these generated scaffolds, and depending on the problem, we get a variety of different structures, but in general, we find that our EvoDiff seek proteins are typically more novel. And we're coming to the end of this a little bit early, but this is totally fine. Essentially, today we've presented this idea of, how can we take sequence alone and generate new proteins with new functions? We've shown a few examples of plausible unconditional generation. We've looked at how we can scaffold these motifs in functional space. And also, we're moving forward and interested in kind of the reverse of this question. How can you come up with a desired function and then design a new sequence that corresponds to that? And with that, I just want to shout out the whole team at Microsoft Research. This is the entirety of the BioMel group, many of these who are authors on the paper, and just want to take a moment to plug that we're hiring. So if anyone is looking for a job or going on the job market, we currently have 2 postings out for a senior applied scientist and an applied scientist too. Feel free to reach out, and I'm happy to talk more about both of those positions. And with that, I'm happy to take any questions.
All right, well, let's thank Sarah. It's a really fantastic talk, and we'll open it up to questions.
Oh, okay. I'm going to stop sharing, but happy to share again if we need to do that so I can see.
Hi, Sarah. Thank you for the talk. I was wondering, so the actual model, when at inference time, you use a sequence to generate a new sequence, right? You don't use any sort of functional kind of keywords to generate the sequence.
Yeah, okay, so I think what you're referring to is this conditional part, correct? We're just using information from the sequence to generate new parts of the sequence. The only context provided is in the parts that we don't mask out in this case.
I see. So when you're using the data and you're saying like many organisms, many functions,
Yeah.
is the idea that the model hopefully learns the functions by itself without like does it what exactly what does function mean, I guess, in this case?
Yeah, I think a good example of this is maybe our MSA example. So when we conduct a homology search, I'm not going to pull up my slides again. When we conduct a homology search, we're looking for sequences that are functionally related, right? And so our MSA contains something about the shared function of those sequences. So when we mask out just the query sequence and we say, okay, you've seen a bunch of examples of models with shared functional properties due to the construction of this MSA. If I give you part of the MSA, can you like fill in the blanks of what might be there? Just based on the fact that you know that these proteins are related. And that's kind of where the functional part comes in. I think there's a lot of information, which what you're getting at is to be added in terms of like, can you include like Go annotations or something about like the language function of this that's critical to doing that. And I think that's like a really interesting thing to explore. But yeah, just for now, the context that's being passed is like, have we learned a relationship via the MSA?
I see. Okay, so when you're doing the MSA search, you also make sure that the functional group is similar.
Yes, correct. Yeah.
Okay. Thank you.
More questions?
Hi, Sarah. That was a really cool presentation. Thank you so much for sharing that with us. I have a couple questions about the generation of disordered sequences. I think that's really interesting. So are there currently other tools that are able to generate realistic disordered sequences?
Yeah, people have used, oh, the name is slipping my head right now. There's another MSA-based homology tool that tries to construct new IDRs based on the identities of similar to like a POTS model, I think, but more learned. So it'll try to construct an IDR based on that. I don't know of any generative deep learning models that can do this, but you know, it's a big field and I could have missed something. But there are some tools that can do it based on like a constructed MSA.
I see, thanks. And what would be some applications where you'd want to generate a different disordered region?
Yeah, I mean, a lot of disordered proteins are involved in diseases. So, you know, in developing drug targets, that might be interesting. Yeah, I think in general, like anything we are interested in developing new proteins for an IDR falls in that category.
Cool, thank you very much.
I had a quick
Hi,
question.
Sarah.
Oh, go ahead, please.
Yeah, hi, Sarah. Very good talk. I have two questions. The first question about the motif scaffolding. In your talk, you said the motifs are randomly put somewhere in the sequence, and actually the motif have multiple fragments. Whether when you put it away, whether the order of the fragments in their primary sequence will also be changed, or they will always keep the same?
Yeah, this is a good question. So what we did was we would take, these motifs were defined in the RF diffusion paper. And so in our, you know, thought process, the simplest way to do this was to take the motif and the spacing between them as being the same. So not changing the spacing between them. And then just scaffolding something of various size around it, randomly placing the motif in the sequence. We did that for consistency across the 17 problems. However, I don't think it's out of pocket to think that you could change the distance between like the broken motifs and, you know, increase the size there, or keep it, you know, fixed. Yeah, so I think to answer your question, it's possible to scaffold them in other ways. We did it to have like a robust method to evaluate all 17 problems.
Okay, good. Thank you. And the second question, when you generate the sequence, it is stepwise, once and once. I want to know what determines the order of decoding those sequences.
Yeah, it's random. So we just randomly decode the order. similar to how it's trained.
Okay, thank you.
Yeah, I had a question. In the diffusion denoising framework, you're adding either the masked tokens or you're mutating a residue at a time. And I'm curious on how, if there's other choices that you could have made here and how you settled on these specific ones. In particular, I'm thinking about increasing the insertions or deletions. That's often a very common evolutionary change that you could happen in DELs. And also, if you could picture that you had a residue that's interacting and you mutate one side of it, that could be, you could get to a place where it's not a very good sequence. So I'm curious if you ever looked at coupled sequence moves where you're changing several positions at a time in order to sort of maintain functional motifs that are kind of embedded in the sequence space. So picturing there's kind of some, large set of all the different moves, but some of them are learned in kind of the way that you could picture making a, sort of the state space is not just on the orthogonal directions, but kind of in the sort of non-orthogonal directions.
Yeah, this okay, this is interesting. Yeah, I haven't thought about that specifically, but I think there are a few. Okay, I'll start with the rationale of why we picked these three methods was that when we started the project, these were the discrete diffusion methods on our radar. And it was a while ago, and the field has obviously been evolving really quickly. I think when you think about things that are maybe noise and not a fixed length setting, my mind immediately goes to maybe those embedding diffusion type models where you're diffusing over an embedding space or something like that. That would be really interesting to explore. And then the third is this, the idea about the indels and deletions. In our EvoDiff MSA model, we are able to include gaps, so insertions, through the nature of how those MSAs are encoded. So we get the gap token and we can just include that in our schemes, which we do. We just have it mutate to that or mask those out. And then at inference time, we see that it can actually generate those within the model. I think I gave a really quick overview of one case where we saw that, but I think there's a lot to explore there. Also in terms of, I think using that MSA-based model to make smaller enzymes seems like a really interesting application of that. But these are all interesting questions, good points. It would be very cool to look at kind of an embedding-based diffusion model where you aren't stuck with a fixed length. I think that's kind of the limitation of these and how they were developed, but I think I'm not 100% sure. I'd have to look at the math, but that would be my intuition. I'm sure I don't know if you have other thoughts on that, but.
No, thank you so much, Liz. More questions?
I have a question. I may have missed it. Oh, by the way, great talk. Thank you so much for coming and doing this. Really appreciate it. But so I had a question when you were doing the comparisons against RF diffusion. So as you mentioned, RF diffusion is a structure-based backbone generator. But because of that, when it's doing the generation, it doesn't produce a sequence. So I was wondering how you got the sequences to do the comparison. Did you use the same method you used, ESM-IF, or protein MPN, or something like that?
Yeah, we did. I don't remember which ones made it into the fun. We compared both ESM-IF and protein MPNN for all of the things we did in our paper. I don't remember which ones made it into our final regular figures, but for the comparisons on their embedding spaces, I'm pretty sure we just used a consistent method for both. But we have these calculations using both methods. There was no rhyme or reason to picking. Actually, there was a slight, we ended up going with the ESM-IF, I remember now. And it's because they seem to have greater differences between if you look in the SI, the ranges of predicted. So protein MPNN, the perplexities might be between 3.2 and 3.6, whereas ESM-IF would give us 11 versus 13. So I think that's why we ended up going with that for a lot of the comparisons. But I do think for all the sequence comparisons, we used ESM-IF. Yeah.
Awesome. Thanks.
One of the really striking things about your work is being able to generate so much more diversity in the sequences compared to these other methods. But I'm curious if in other diffusion models, there's often a knob where you can dial it between low perplexity and greater diversity.
Mm-hmm.
So I'm curious if you had a similar type of knob in your model where you could trade off balancing diversity for, like, for instance, in your when you're doing the motif scaffolding, maybe you just want to make like very conservative mutations to try to like stabilize or enhance an enzyme. Or if you're generating like something that's totally new, maybe that's a very risky thing to try. So you might want to dial back that diversity if you could somehow get better, perplexity or see perplexity in your metrics.
Yeah, this is a great point. I think so. For the purposes of this paper, I think we just wanted to not turn knobs and like see what comes out of it, what it can do. I think when we are going and we're thinking, if anyone's we're hoping to like expand this and work on more applied projects. And I think that's a case where you can then fine tune like what it is that you want out of these models. Like in that case, we're not like for each problem trying to optimize the number of successes. It's more like, oh, what's it capable of? And then moving forward, it's like, oh, we have this problem that we would love to like synthesize or take to a lab. Like, how do we make the best version of that? And I think that's a case where more knob turning seems more relevant. But yeah, for these, we didn't like play with temperature or sampling or anything like that, or yeah, nothing crazy.
More questions?
I have another one if you don't mind.
Yeah.
So I know like in hearing people talk about results they've gotten from RF diffusion, I've heard discussions of like how it tends to create kind of idealized folds and proteins that tend to behave very well when expressed. So I'm wondering, maybe while those may behave well when expressed, it's not encapsulating the full like diversity of natural sequences. Do you think EvoDiff would do that better, or is there any reason to believe it would also be doing kind of idealized cases?
Yeah, we're looking into this exactly right now. The rate at which we can, run experiments is a lot slower than an academic lab. So sure. I'm hoping to have an answer on this soon. I shouldn't undermine this. There's like its own set of difficulties that come with expressing from just a sequence and like not a structure. I think it's a little bit more difficult. So yeah, I don't know. I don't think a one-to-one comparison is fair here, but I am hoping we'll know a little bit more about this in our like round of experiments that we're running right now. And hopefully this will be out. Yeah, soon.
Thank you. Looking forward to hearing how it goes.
Yeah, thanks.
Thank you.
One of the challenges with discrete models in general is that oftentimes they're really tricky to train. The loss signal is discretized in a lot of ways. And so I'm curious if you could give any color on like challenges that you encountered in trying to get this model up and working, and sort of what parameters you thought were sort of the most sensitive or most important. I know in a lot of the diffusion models, the temperature schedule is super critical. So I'm curious if you like ran into issues that you were able to solve and sort of give any sort of experience on how this went, and when people are trying to do sort of similar models for like rotamer side chains or something like this that might also be discrete.
Originally this project, we really were focused on the D3PM scheme, which is a much more complex problem than the OA-ADM. The OA-ADM is just like a reweighted loss at every step over mass token. So it's like shockingly easy to implement. The D3PM is a little bit more complicated in that, yes, you need to have a schedule that, so there's evidence that like the cosine-based schedule, the patterning of that schedule can lead to better results. And I think we hadn't thought too deeply about that. So we tried to just develop a schedule that was mutating like almost 1-to-1 at each step over 500 time steps. And we ran like a couple experiments, but we just settled on something that was close to linear. And there is like a lot of development in these methods, but we ended up just going with some empirical schedule that kind of tapered like a logarithmic scale to try and get that pattern over many different size protein sequences. And it seemed to work well, but I do think had we tried to mimic a shape of like a cosine, we would have gotten maybe better results. That's like an interesting thing I think we could experiment with, because the results of our D3PM model didn't scale as well as we thought it would. And in terms of like, the implementation of that math was a little bit tricky, just in that, I'm like a methods developer, so I don't know the thought that went into developing this, but the math behind it is a little bit tricky. So if anyone's interested in developing that, I'm happy to get a little bit more into how we did that. I also don't think our implementation is the most efficient because it has like 3 parts, and I I think it could be faster if anyone's like an ML engineer and interested in making that faster.
Amazing proof of concept. So definitely.
Yeah.
I guess sort of a follow-up. In a lot of these architectures, we see sort of relatively stable scaling laws. So like performance as the
Yeah.
data increases, the compute
Yeah.
increases, or the architecture increases. And I'm curious if you, to the extent that you've tested this out, where
Yeah.
you sort of see those scaling laws falling in. Like, is this, you could just feed it infinite amount of sequences and you could drive the loss to zero, or is it,
Yeah,
are you sort of
this
hitting
is
some sort of like ceilings on some of these dimensions?
yeah, this is a thought we've had as well. Our thought is it'll probably the way of training the order agnostic autoregressive model is also much less efficient than a plain autoregressive model, because you're like not evaluating everything at once. So it scales a lot slower than that model. And we only have 2 data points right now. We only trained a 38 million parameter and a 640 million. So yeah, it'd be really interesting to see how these things scale. It's something we're looking at, and yeah, hopefully, I don't know when we'll have the answer, but it would be an interesting answer to have. And I should add, I think our models are, EvoDiff seek, this is not something we've been talking about, but it is a little bit undertrained. I mean, we're limited by how long we can train these models, and it certainly, like, these can be trained longer and we can get better results, but yeah, it's a matter of like how much resources we have. And yeah, it would be cool to continue exploring these at scale. Hopefully we've like given evidence that we're allowed to explore this.
For sure. More questions?
Yeah. And I should add Heart paper from Kevin Yang. He explored some scaling. Our backbone is quite similar to his, so we could expect something kind of in that vein.
Nice. That's great to know.
I have a quick question about the architectures used. So if I remember correctly, in EvoDiff Seek, you said you used a convolutional neural network rather than like a transformer that you used with the MSA mode,
Yes.
MSA version. I was curious if you could talk about why you chose for a convolutional network for Seek rather than like a transformer-based network. Yeah,
we wanted to just make some comparisons to Kevin Yang's CART model. Originally, when this project started, we just wanted to make some comparisons. It was kind of the easiest out-of-box thing to use. It would be, I think, really interesting to see how, or if, a transformer model would affect that. The original reason, and this is something Kevin explored in one of his earlier papers with the CARP model, was that he was able to simulate or model longer protein sequences with the CNN compared to the transformer. And so that was like the initial motivation for that. But yeah, it would be interesting to explore like what the results of a transformer might be.
In your introduction, you had this really interesting observation that the structures themselves are kind of fixed, whereas sequences are functional, right? They do things. And the intrinsically disordered is a really excellent example. But I'm curious if it maybe you saw in other cases sequences that had sort of intrinsic either fold switching or maybe sort of indications of sort of breathing or dynamics or something that would be hard to capture with a fixed structure beyond just say like an intrinsically disordered region.
Yeah, this is my personal MO. My background is in molecular dynamics, and yeah, I'm really curious. I don't know if we found examples of this because we weren't looking for them, but I know that they exist. Like, I think dynamics is a huge part of this equation, and like how, yeah, proteins are dynamic molecules. I think to not consider that as being part of the problem is like huge, but it's also a really complex problem. So
okay,
I forgot what the question was. I got excited about dynamics. Yeah, but yeah, if there's any, yeah, this is like a long-term interest of mine, is like how you can combine these two modes and like maybe create better models.
I think we have
I don't think I answered your question. I forgot what your question
was. No,
was.
Absolutely.
Just talking.
There's so many cool questions that this model opens up, so I'm really excited to see where it goes. Any other questions?
I just had a question about the Prote5 embeddings. So for the test proteins, there seems to be like a region on the left that the models can't replicate or reach. Is that like, do those proteins share like certain properties?
Yeah, this is something that keeps, coming up and is something I need to look into as to like what those proteins are. It does seem like our autoregressive model can get there. So I wonder if it's just, a matter of longer training times to get there or what. But yeah, it is interesting. I am curious as to like what those are. I don't have an answer for what is in that region. I'm assuming it's like a functional or type of protein or something.
Thank you.
Yeah.
Kind of bouncing off that real quick. I was wondering, you mentioned that kind of RF diffusion and your model was kind of orthogonal in the way that the proteins, there's kind of success on the two axes there. I was wondering, do you have any insights into which proteins would be better to use with RF diffusion rather than your model or vice versa?
Yeah. No, and yeah, this is a great point. It's the set of things we look at is so small. It's 17 problems, and both of our models can do like beta scaffolding, helix scaffolding around something without context. Oh, I might have to take off in a second. But yeah, there doesn't seem to be some like obvious correlation in why one does better than the other that I've seen. Maybe, yeah, I don't know. I'm hoping someone just reaches out and is like, oh, that's why. But yeah, I don't know. Like the problems themselves, it doesn't seem quite obvious as to like why one is better than the other.
Okay, well, maybe in interest of time, let's thank Sarah for a really excellent talk and really incredible contribution to the field. So thank you so much for coming, and we'll see you in 2 weeks. I think we have another lineup set. So
and really quick, sorry, I forgot to plug. I forgot this is mostly students, but we normally take interns in intern applications. I think in like November. We're done this cycle, but I'm sure many grad students will be around then. And yeah, if you're interested, feel free to reach out to me or anyone on the team.
We have a Rosetta jobs mailing list, so maybe if you want to post something to send it there.
Okay.
The Rosetta community is pretty good, and there's a collection of people. So if you're not on that list, you can join too if you're looking for a job too. So.
Okay. Cool. Awesome. Okay. Thank you so much.
Okay. Thank you so much. Take care.